Dear listeners, welcome to another episode of the podcast about enzymes and molecules. I am Roberta Foresti, Professor of Biochemistry at the University of Paris-Créteil and researcher at the Mondor Institute of Biomedical Research in France. In the first two episodes, we discussed heme oxygenase and its products, carbon monoxide and bilirubin, and today we are coming full circle with this subject by specifically examining the substrate of heme oxygenase activity, which is the heme molecule. Heme is very abundant in our body and several proteins contain it, usually bound to their active site and supporting the function of these proteins. Examples of proteins include hemoglobin, present in red blood cells and responsible for transporting oxygen to all tissues and organs, or organelles such as mitochondria, which are rich in heme-dependent proteins that are indispensable for the production of cellular energy via the respiratory chain. Iron, which we acquire through nutrition, resides in the center of the heme molecule and is the critical element that supports its function. Despite its essential role in the efficient working of our organism, there are certain situations where heme causes damage. Today, we have the pleasure of hosting a dynamic scientist that has focused her research on the study of heme and iron in the context of hemolytic diseases, such as sickle cell disease. I am delighted to welcome Dr. Francesca Vinchi, an Italian scientist working in the USA. Dr. Vinchi is assistant professor at the Lindsay Kimball Research Institute, the research branch of the New York Blood Center in New York, USA. She's a molecular biologist by training with a PhD in biomedical sciences and human oncology from the University of Turin, Italy. Before moving to the USA, Dr. Vinchi trained for 5 years as a postdoc in iron biology at the University of Heidelberg and European Molecular Biology Laboratory EMBL in Germany. At the Lindsay Kimball Research Institute, she is currently head of the Laboratory of Iron Research, where she investigates the detrimental impact of heme and iron in sickle cell disease and other pathologies, all marked by an abnormal heme and iron metabolism. Today, Dr. Vinchi and I will talk about different facets of heme biology, why heme is crucial in normal physiology, and why it exerts a deleterious action when it is not tightly bound to a protein. We will further examine the modulatory role of heme in diseases characterized by excessive free heme in circulation, and how endothelial cells in vessels and specific immune cells called macrophages adapt their function in response to heme. We hope you will enjoy our conversation with Dr. Vinchi and learn more about the heme molecule and its effects in human pathologies. As an Italian scientist living in Paris, interviewing an Italian colleague working in the USA, I am sure our listeners will not mind if we greet each other in Italian. Ciao Francesca, siamo molto contenti di averti con noi oggi sul podcast. Come stai?
Ciao Roberta, è un piacere registrare questo podcast con te. It's really a pleasure to be here with you to record this podcast.
Thank you, Francesca. Heme is the subject of our conversation today. Can you tell us why this molecule that is so crucial for life can become dangerous in specific pathologies?
So yes, definitely heme is a very interesting molecule. We can consider heme as a molecule with really a double-edged sword natures. As Roberta mentioned very well before, is of course an essential molecule to living organism because it's an essential cofactor for a lot of hemoproteins and covers a number of biological function, including oxygen transport, respiration, drug detoxification, and also signal transduction. As Roberta said, for example, in hemoglobin and myoglobin, it's needed for oxygen transport and storage, whereas in other molecules such as cytochrome is involved in electron transport, energy generation, and chemical transformation. Again, in catalases, instead, is needed for hydrogen peroxide degradation. And but beside this, also heme has a lot of additional function, including protein synthesis regulation and control of gene expression. Although on one side, so its function is really essential, as we said, it has a particular nature because on the other side, when heme is present intracellularly or extracellularly in a cells, and when its levels are not finely regulated, heme can become toxic. We indeed need to remember that free heme is a source of redox active iron. So it's the tetrapyrrole that coordinates inside an iron atom, and the iron atom is highly reactive, can participate into a chemical reaction which is called the Fenton reaction that leads to the production of free hydroxyl radical, also called reactive oxygen species, that can interact with many other biological molecules, including lipid membranes, proteins, nucleic acids, and therefore damage this biological component and lead to cell damage, tissue injury. Beside the ability to trigger ROS formation, we have to remember that heme could also be toxic because of its own nature. It's a quite lipophilic molecule that can easily intercalate into membranes. Therefore, it can damage directly the phosphodilipid bilayer in the cells and within organelles, and cause enhanced membrane permeability and cause also cell death. So this is all a number of mechanisms that we have to remember might occur in presence of heme excess. And we know there are multiple pathological conditions that are associated with heme overload, where free heme can cause the toxic effect that I mentioned before.
Excellent. Very good explanation. Very wide. So really, there is a wide variety of functions for heme that make it, as you say, essential, and at the same time, we need to be able to control it because it has this pro-oxidant activity. So what is the prevalence of diseases characterized by hemolysis in the population? So hemolysis, you are going to explain to us what it is, and probably you will tell us a little bit more about these diseases in the population.
Exactly. Yeah, I would like to go through the type of hemolytic anemias where excess free heme might be generated. So there are essentially two types of hemolytic anemias: the inherited hemolytic anemias. These usually are caused by a defect that occurs in the red cell itself, in the sense that one or more genes that control red cell production or the production of some component of the red cell don't function properly. So usually in this condition, what happens is that the cells are destroyed prematurely, and this destruction might occur usually within the spleen, where macrophages are present and therefore pick up in an accelerated manner this abnormal red cell and eat them, remove them from the circulation. Or when the red cell is particularly altered in its shape and structure, the destruction can also happen intravascularly within the vessels. Usually this type of inherited hemolytic anemias include sickle cell disease, for example. Sickle cell disease is caused by a single point mutation that happen in the gene that encode for beta-globin, which is an essential component of hemoglobin. And basically this mutation makes in a way that an abnormal form of hemoglobin is generated, which is very prone to polymerize. So polymers of hemoglobin are generated that cause the elongation of the cells. So that's why the sickle red blood cell becomes very elongated and rigid in the shape. And as having this abnormal structure is particularly prone to rupture, to breakage into the circulation, and also to occlude small vessels because of this elongated shape. Another disease where a globin gene is altered is also beta-Thalassemia, for example. In beta-Thalassemia, we don't really have a mutation in the globin structure, but rather a reduced or lack of expression of the beta globin. And this causes, on the other side, an accumulation, an imbalance in the other globin that create hemoglobin, which is the alpha chain component. This alpha globin tend to precipitate, and also in this case, creating the so-called hemicron, that is a composition of alpha chain and also iron. The red cell becomes very fragile, eventually very prone to rupture, and also removal, prone to be removed from macrophages in the spleen. Other type of also hemolytic anemia associated with alteration in the red cell shape are also, for example, spherocytosis or ellipsocytosis. And we have also to remember some deficiency in enzymes that promote hemolysis of the red cells, such as the pyruvate kinase deficiency or the glucose phosphate dehydrogenase deficiency. The second type and second class of hemolytic anemias instead include a number of anemias that is not caused by an alteration in the components of the red cell itself, is rather caused by external factor from the red cells, which could include the presence in the circulation of antibodies directed against the red cells, and this happens when autoimmune disorders are present, for example, so the red cell tend to break in the circulation because of the presence of these antibodies. Also, a number of medication and drugs can promote an accelerated hemolysis of red cells. And we also have to remember the hemolysis associated with infection. Indeed, malaria and babesiosis often cause this type of acquired hemolytic anemias where red cell breaks in the circulation. So, in terms of prevalence, worldwide anemia is pretty diffuse. So across all ages, there is a 25% global prevalence of anemia. And we have to remember that approximately 5% of all anemias is represented by hemolytic anemia. Definitely the most diffuse type of hemolytic anemias are represented by sickle cell disease and thalassemia. And round about 5% of the world's population really carries traits for this type of hemoglobin disorders, rather sickle cell disease or thalassemias. While thalassemia is more common in Asia, in the Mediterranean areas, and in the Middle East, instead sickle cell disease predominates mostly in Africa. And about 300,000 babies each year are born with this type of severe hemoglobin disorders. And even just in the USA area, there is a population of about 100,000 sickle cell disease patients. And because of migration also from Africa into different areas of the world, we have to remember that thalassemia, sickle cell disease have become nowadays disease diffused in a region where before doctors were not used to see these patients. So, of course, the societies involved in the learning and diffusing the knowledge on hematologists are nowadays trying to activate and this program to make doctors and scientists more aware about this type of disease and to expand the scientific research that is focused on these hemolytic disorders.
So you are telling us that these diseases are actually quite common, more common than maybe is appreciated normally. And that I think you raised a very important point, the fact that with migration, people will diffuse, will take the genetic predisposition with them in the new areas, and therefore there isn't really the expertise in terms of healthcare, in terms of knowledge about the disease and how to take care of the disease, how to treat the disease. So this is also another important issue that perhaps is not yet fully appreciated, I would say. So in our blood, we have specific proteins that have the capacity to bind heme. and scavenge it so that it cannot circulate freely as an unbound molecule, such as, the molecule that we would normally find in these diseases. What goes wrong in diseases such as sickle cell disease? Why can these proteins not protect us anymore from heme-mediated injury?
Yes, Robert, as you mentioned, basically, usually in normal condition, our organism is really equipped with a number of systems that allow us to prevent this type of heme toxicity, so the reaction of the of the iron containing the heme molecules. And basically, these systems are in part intracellular system, in part also extracellular system, and these allow the control of intracellular and extracellular heme levels. As you know, and since you studied extensively along the years the function of hemoxygenase 1 and 2 are essential heme catabolizing enzyme that controls heme levels within the cells by degrading heme and avoiding its accumulation into the cell. Instead, extracellularly, we are equipped with hemoglobin and heme scavengers. The specific hemoglobin carrier is called haptoglobin. And this is a molecule that, when complexed with hemoglobin, delivers it mostly to macrophages that express CD163, that is the specific receptor for the hemoglobin-haptoglobin complex. However, when eventually hemoglobin levels in the circulation are elevated, haptoglobin can be saturated, so its capacity is overwhelmed. And this leads to the generation of free hemoglobin that eventually undergoes oxidation into a methemoglobin. And in this situation, at this point, it's important the function of a secondary scavenger, which is the specific scavenger for heme, which is called hemopexin. Hemopexin, by scavenging heme, mediates its delivery to macrophages and the pathocytes specifically that express LRP1, also called CD91, which is the specific receptor for the heme hemopexin complexes. And basically, by promoting heme delivery and detoxification in these cells, hemopexin really acts as kind of antioxidant, so prevents heme-induced oxidative damage, cell death caused by heme, and reduce heme toxicity by really 80-90%. So, in general, therefore, the function of these two scavenger, haptoglobin 4 and hemopexin, is critical in acute condition. For example, when a small hemorrhage occurs in the body and hemopexin are around to buffer the excess of hemoglobin and heme which are released from the red cells. Their role is also extremely important in more chronic anemias, where, of course, the function of haptoglobin and hemopexin becomes even more important because the abundance of hemoglobin and heme that are released is even higher. However, there is a point where, unfortunately, the amount of haptoglobin and hemopexin is not sufficient anymore. We have to think that these both haptoglobin and hemopexin are acute phase protein. They are released during condition of infection and inflammation, because in this condition, hemolysis occurred to some extent. So their production basically minimizes tissue injury and facilitates tissue repair when heme and hemoglobin are released. However, their production is also balanced by basically their utilization. In chronic hemolytic anemias, which are pathological conditions, the utilization of these proteins is so chronic due to the chronic release of hemoglobin in the heme that their level, their amount in the circulation of hemolytic patients basically drops progressively. So their production is not sufficient compared to their utilization. And it's exactly at this point that the organism remains, let's say, unprotected, because a level of heme, of free heme, is generated that cannot be bound anymore by hemopexin, which is then insufficient, and therefore heme toxicity, heme-mediated damage might occur.
Excellent. So it's, I didn't have this very clear in mind, but haptoglobin and hemopexin work in tandem. First, there is the haptoglobin scavenging the hemoglobin, and if this is not enough, we then have the emergence of hemopexin, which will try to counteract the heme toxicity by scavenging directly the heme molecule.
Exactly.
So in a way, if we could prevent hemoglobin oxidation and loss of heme, we might not really need hemopexin, but obviously the fact that it's there, it means that it's very difficult to prevent hemoglobin oxidation.
Yes, also because in the circulation, changes and oxidation are events that occurs more frequently. There is less control over the oxidation processes compared to the intracellular space. Therefore, oxidation is an event that can occur. So when methemoglobin is generated following oxidation of hemoglobin, hemopexin is the ultimate scavenger that has an antioxidant and protective relevance. Yes, definitely.
We understand that when there are not enough heme-binding proteins, cells and tissues will be exposed to the excess heme. And this, in turn, will trigger specific responses. And a lot of your work has really been dedicated to studying this aspect. You have shown, for example, that macrophages in the liver are particularly affected by heme. Can you tell us about your findings and what are the implications of your results?
So, yes, definitely macrophages are cells particularly sensitive to free heme. And I focus a lot of my research in really understanding how heme can change the phenotype and the function of macrophages, let's say, in the last decade. We have to remember that macrophages are key cells of the immune system, and they are all marked by remarkable plasticity. And for plasticity, we mean a kind of adaptation which reflects substantially the ability of the macrophage to integrate, respond, and adjust to a number of microenvironmental stimuli. So thanks to this type of plasticity, when something wrong happens in the body, macrophages can acquire a different functional phenotype that allows the restoration of homeostasis. And they do this through regulating inflammatory or anti-inflammatory processes. Therefore, we have been aware that plethora of phenotype of macrophages can occur in different situations, going from an extreme situation where basically macrophages acquire pro-inflammatory hallmarks and phenotype. They produce elevated pro-inflammatory cytokines, and these macrophages usually are implicated in fighting infection and microorganisms and stimulate the activation of other immune cells. On the other extreme of the spectrum, macrophages can also acquire an anti-inflammatory phenotype, so they can produce anti-inflammatory cytokines, and usually this type of cells are mostly involved in tissue repair and immunoregulatory processes. Besides their immune role, we have to remember that macrophages are also key players in the heme and iron homeostasis, because as I mentioned before, macrophages can clear senescent red blood cell. They remove these red cells when they are aged, and they degrade the hemoglobin-derived heme, and they can also uptake hemoglobin and heme bound to their respective scavenger, haptoglobin and hemopexin. However, while the scavenger-mediated uptake and also erythrophagocytosis are basically regulated processes that allows to control and limit heme and iron accumulation, what happens in hemolytic condition is completely different. This regulated uptake is now overwhelmed, and now macrophages are exposed to free heme and accumulate a lot of heme and iron. What we were interested for a long time is exactly to understand how this uncontrolled heme uptake by macrophages could change also their immune response in terms of inflammatory or anti-inflammatory pathway. And basically, what we have discovered, we as well as another group, is that heme really represents a major environmental stimulus that, upon a chronic hemolysis, determine a switch, a change of the macrophage phenotype. So the macrophage acquire a highly pro-inflammatory phenotype, start to produce inflammatory cytokine and reactive oxygen species. And this happened because heme acts as an alarmin, in the sense that alarms our body that something is wrong, that something is going on, that a damage is occurring. And by allowing this type of sensing, heme activate the macrophage that send this signal around the body and activate an immune response. So this is exactly what we have found, for example, analyzing mouse. So we have observed that in the liver, macrophages are chronically activated in an inflammatory direction. They produce a lot of cytokines. And on a mechanistic level, this depends on the ability of the heme to activate a specific signaling pathway, which is started by the activation of the toll-like receptor 4. And on the other side, it also depends on the production of reactive oxygen species mediated by heme. So when we block either TLR4 activation or ROS production, we no longer see immediate activation of macrophages into a pro-inflammatory direction. Very importantly, we have to consider that while in an acute condition of hemolysis, this is a temporary process. So basically, the macrophage is activated temporarily towards a pro-inflammatory direction by heme to reestablish then homeostasis. Obviously, this process became chronic when hemolysis is indeed a chronic event, as in sickle cell disease and β-Thalassemia. So the heme-induced pro-inflammatory activation it's substantially, essentially never-ending and promotes a condition of chronic sterile inflammation, which is indeed the hallmark and characterized disease as sickle cell disease. And in this condition, as you might understand, the chronic inflammation ends up being a detrimental process that also contributes to tissue damage, in a tissue like the liver, also to aggravation of hepatic fibrosis, and also organ damage and eventual failure. So we have thought that administering hemopexin, for example, to scavenge could basically prevent or limit to some extent this immediate pro-inflammatory activation of the macrophage. And we have tried in mouse model of sickle cell disease to administer hemopexin as a therapy. And indeed, we have found that we could reduce the overall inflammation by scavenging heme and reducing this pro-inflammatory response of the macrophage. A second aspect where we focus more recently is, let's say, really understanding how heme could impact the function of the macrophage. So not only in terms of inflammatory or anti-inflammatory response, but also understanding how essential function of the macrophage could be altered, and we focus specifically on efferocytosis, which is the process of removal of apoptotic cell by macrophages. We thought this process is crucial because in sickle cell disease, the sickle-shaped red blood cells are prone to remain stuck in the vessel, as we mentioned before, and to promote vaso-occlusive crisis. This crisis basically, by reducing tissue oxygenation, could cause cell apoptosis, tissue damage. So the removal of dead cell could be essential in this condition. And what we have found is that using in vivo model and in vitro approaches is that basically heme is capable to change the function of the macrophage, in particular impairs the efferocytic capacity of the macrophage. The macrophage is no longer able to eat efficiently apoptotic cell, to clear these cells. Why this is important? This is also important not only to reduce tissue damage by clearing the apoptotic cell, but it's also important for a secondary aspect. The removal of apoptotic cell usually triggers a switch of the macrophage towards an anti-inflammatory response, and it starts an anti-inflammatory mechanism that is aimed to repair the tissue damage. So what we have observed is that basically Heme, by blocking the removal of apoptotic cell, prevents also the activation of this anti-inflammatory mechanism in the macrophage, which leads to a sustained inflammation and blocks the anti-inflammatory pathway and tissue repair. We found that, again, also the block of efferocytosis is due to an activation of the TLR4 pathway. And this is also dependent by the ability of the Heme to reduce two important transcription factors, which are called PGC1 alpha and PPAR gamma, which regulate the experts that are responsible for the uptake of apoptotic cells. So Heme, by reducing with this transcription factor, suppresses this receptor that recognizes apoptotic cell, and overall this leads to a reduction in the efferocytic process. And once again, we have proven that administering hemopexin as a therapy or trying to stimulate the induction, the expression of PGC1 alpha and PPAR, the transcription factor implicated, we can ameliorate macrophage function. We can render these cells more capable of performing efferocytosis, and therefore this leads to a better resolution of inflammation and of tissue damage, and a nice clearance of the dead cell in the tissue, such as the liver, which overall leads to decreased fibrosis, decreased organ damage. Why the clearance of these apoptotic cell is also important is because it allows the removal of self cells. So you might imagine, and what our results have demonstrated is that when Heme block efferocytosis, it favors autoimmunity in the sense that autoantibody and autoimmune response can be mounted against the apoptotic cells that are not cleared, that are not removed. And while this has gone underappreciated for a long time, we have progressively observed that since nowadays patients with sickle cell disease lives longer because they have better therapies, autoimmunity has never been a thing, never been really looked into. But lately, in these last decades, we have been observing that also patients with sickle cell disease are more prone to the development of autoimmune diseases. And so we truly believe that Heme, by blocking the efferocytic capacity of macrophages, could contribute to this type of autoimmune disease also in sickle patients, for example.
Many different consequences of Heme, excess Heme, and they really go beyond the classical thinking of promoting ROS and having an effect on the inflammatory profile of macrophages. Very interesting. And I find also that it's very interesting that there is a population of macrophages that are literally patrolling blood vessels and remove damaged cells and debris from the vasculature, helping to maintain and protect the vascular tissue from injury caused by abnormal red blood cells, for example, in sickle cell disease. Can you explain how they can do this from a mechanistic point of view?
Yes, these cells, the patrolling monocytes, and indeed the particular cells, they crawl along the luminal side of the endothelial cells. So then they are almost considered as a kind of accessory cell of the endothelium. And they were implicated basically in the phagocytosis of this debris that derives from damaged endothelial cells. And particularly lately, a group of colleagues at the New York Blood Center indeed describe a novel subset of these patrolling monocytes in sickle patients that are characterized by an elevated expression of the heme catabolizing enzyme oxygenase 1. And basically, what my colleague has been able to show is that this patrolling monocyte subset is capable of clearing specifically endothelial cells that have been damaged as a consequence of hemolysis. And so by picking up these endothelial cells damaged by hemolysis, this also leads to a concomitant upregulation of hemoxygenase, eventually to degrade the heme excess containing these cells. This process is dependent on the expression of ICAM1 and also the presence of phosphatidylserine on the endothelial cells. And but importantly, what more recently they have been able to describe is that not only these endothelial cell damaged by hemolysis are picked up by this subset of patrolling monocytes. Beside this, also really sickle red blood cells that stick to the endothelial cells are removed by these patrolling monocytes. So they have a double function, both removing damage and sickle red cells that stick to the endothelium. And in this way, these macrophages, these monocytes, seems to have a protective role also against the pathogenesis of vaso-occlusive crisis, because they remove these sickle cells that are prone to stick to the endothelium. Basically, what they observe is that in patients with sickle cell disease, we can indeed appreciate inside patrolling monocytes some red blood cell engulfed material, so really telling us this process is real. And mice, for example, that lack patrolling monocytes are particularly sensitive to damage caused by hemolysis at the level of the endothelium, since they are not able to clear these endothelial cell damaged by heme. And they seem to be more prone also to the development of vasocclusion, since also sickle red cells are not removed anymore by these patrolling monocytes. Even depleting in the sickle mice patrolling monocytes have allowed to observe an increased frequency and exacerbated vascular studies, is really telling us that vasocclusion occurs more frequently. Instead, using strategies that allows us to amplify, to extend the mass of patrolling monocytes, it seems to reduce vasocclusion and organ damage in sickle cell disease. So this is a very interesting aspect that was totally unexplored since some years ago, and suggests that basically this is beside hemopexin and haptoglobin, these patrolling monocytes are an important line of defense against hemolysis-driven endothelial damage and pathophysiology of vasocclusion. So not only heme and hemoglobin scavenger, but also this type of cell have really a protective function. What my colleagues have also been able to describe is that, importantly, patients that with sickle cell disease that had a recent episode of vasocclusion have a decreased number of these pathologic monocytes expressing high hemoxygenase, suggesting that probably misbalances in the number of these pathologic monocytes with expression of hemoxygenase could rather predispose or not to the to this vaso-occlusive crisis as a result of limited clearance of these endothelial cells or these sickle red cells. So this is really a mechanism that determines the balance between probably developing more or less vaso-occlusive crisis. Mechanistically, they have also described how these patrolling monocytes basically tend to differentiate from classical monocytes. So recently they have shown that hemolysis drives both the production of the colony-stimulating factor and also the chemokine CCL2. And while colony-stimulating factor usually trigger the differentiation of classical monocytes into patrolling monocytes, instead CCL2 drives the transmigration into tissue of the classical monocytes and their differentiation into macrophages. So basically, the level of these two factors determines the balance between differentiation into pathologic monocytes or differentiated macrophages. And very interestingly, they've shown that when we block monocytes transmigration through, for example, anti-selectin antibody, anti-P-selectin antibody, we can increase the mass of the pathologic monocytes. So really telling us that usually the differentiation of classical monocytes into tissue macrophages occurs at the expense of their differentiation into pathologic monocytes. And probably we could also use this and leverage this mechanism from a therapeutic point of view to try to reduce vaso-occlusive crisis by expanding the mass of pathologic monocytes and reducing, therefore, the damaged endothelial cell and sickle cell that stick to the endothelial wall.
What you're saying could be a very important explanation for why patients don't all react in the same way to vaso-occlusive crisis, for example. And it looks to me that having all these different protective systems, which are haptoglobin, hemopexin, patholin, macrophages in the liver that have another function. And we can imagine that patients will have different levels of haptoglobin, different levels of hemoglobin, and a capacity, a more or less capacity to develop these helpful macrophages. All these factors will determine the response of the patients and the susceptibility of the patients to the crisis, which happen, but not all of them leave this crisis in the same way.
Yeah, and also probably a difference in the perception of pain, which is indeed also a downstream event that occurs in association also to vaso-occlusive crisis. So modulating all these protective mechanisms having different level, it's a determinant of the crisis and also the pain downstream. So
absolutely, yes. So you described to us very well how these different types of macrophages are reacting to heme and hemoglobin. And there are other cells, which you already started to mention, the endothelial cells in blood vessels that form the internal layer of blood vessels that are also obviously exposed to high levels of free heme. So I assume that also the endothelial cells will have their own intrinsic response, which may be similar or different to that of macrophages. And can you tell us how they respond to free heme and what are the consequences for cardiovascular function?
So yes, we have to remember that indeed in hemolytic patients with sickle cell disease and thalassemia, the presence of endothelial dysfunction, vasculopathy, and impaired cardiovascular system is in general, for certain aspects, a common feature. So really telling us that hemolysis is contributing somehow. And this is to some extent expected because we have to think about that when free heme is circulating in the blood, the vessel wall is the primary tissue that is exposed to heme and to heme toxicity. So in the last decades, we have been focusing and recognizing that oxidative stress, inflammation, also mediated by heme toxicity, contribute to this vaso-occlusive crisis, to the damage at the endothelium, to thrombus formation in hemolytic condition. And in particular, at the time when I was still a postdoc, I was really extensively studying how in sickle cell disease heme was causing vascular toxicity and how hemopexin was important for preventing it by scavenging free heme. And what we have been able, we and other groups to identify is that basically heme release from the red cells is capable to activate the endothelial cells in a quite similar way to what I described before regarding the macrophage. So it activates again the TLR4 signaling pathway and downstream the transcription factor NF-kappa B, and it also leads to the production of reactive oxygen species. And within the endothelial cells, these signaling are required to basically induce the expression of adhesion molecules and also to stimulate the degranulation of small organelles that are called Weber palette bodies that are open up and release their content of P-selectin and also Wallner-Willebrand factor on the surface of endothelial cells. So through all these mechanisms, release of the Weber palette bodies and expression of adhesion molecules, endothelial cells are now more capable to basically interact with leukocytes and also with red cells, especially sickle red cell. And as you might imagine, this mechanism of the endothelial activation and retention of leukocytes and red cell definitely favor red cell stasis along the vessel wall and promote the accumulation of and aggregates of cells that indeed underlies the pathogenesis of vaso-occlusion and vaso-occlusive crisis that is a hallmark of sickle cell disease. Besides this, besides simply the activation of the vascular cell, we have also to remember that hemoglobin and heme definitely change the condition of vasoconstriction, vasodilation of the vessel wall because they alter the bioavailability of nitric oxide. So free hemoglobin definitely scavenge this vasodilator that is nitric oxide and reduce its bioavailability. But also heme, for example, by favoring the production of reactive oxygen species, tends to increase the oxidative consumption of nitric oxide. So it's also in this case less bioavailable. So there are multiple mechanisms via which nitric oxide is rendered less available due to elevated hemoglobin in heme, and this results in the dysregulation of the endothelium and usually leads to a severe vasoconstriction and also hypertension, which can be indeed a hallmark feature in sickle patients. So what we have also found is that indeed, even analyzing mouse model for sickle cell disease, a complex number of vascular event occurs. So sickle cell disease animals are characterized by endothelial activation, vascular dysfunction, a common feature of vasculopathy. But they also have elevated blood pressure, and they really have basically alteration at the heart associated with a compensatory hypertrophy, which is probably mediated by the increased vasoconstriction of the vessels, and they show in general an increased cardiac output due to this compensatory hypertrophy. So what we have been found is that indeed by scavenging heme, we are capable of restore a number of these vascular events. And so when we give hemopexin as a therapy mouse model of sickle cell disease, we see a normalization of a number of these vascular parameters, including the blood pressure, the endothelial activation, and the heart phenotype. So really telling us that, as Roberta mentioned before, not only the macrophage and the activation of the macrophage is a central feature mediated by hemolysis, but that definitely the cardiovascular system is another essential target of heme toxicity.
Fantastic. The message is that a disease with multiple facets, because like heme, it has multiple facets, and probably it's very important that in the treatment and in the care of this disease, all these aspects are being considered: the vascular consequences, the cardiac consequences, the inflammation that may affect organ fibrosis, like for example in the liver that you were mentioning. It's really...
And what I want to add is that now today we have spoken specifically about the vascular feature and the macrophage and inflammatory feature, but the effect of heme has been demonstrated to be much more complex in sickle cell disease, because heme is capable of activating the complements, of activating platelets, also of activating neutrophils. And all these components are definitely occurring in sickle cell disease and determine the pathophysiology of vaso-occlusion. So while my science has been mostly focused on the cardiovascular system and on macrophages, many other studies demonstrated that other cell type and system are activated by heme, and so heme-driven pathophysiology is much more complicated than what we have just been focused today in the podcast. So
yes, thank you for precising this aspect, which in a way leads me to my last question, because what do you think are the fundamental issues that still need to be resolved concerning the toxicity of free heme and iron and hemoglobin?
So I think this is a very difficult question to answer, probably because we have explored so much and discovered so much beyond what we initially thought many years ago to discover. So heme has really many effects, as I suggested before, on many different cell type and system. However, probably we have still been focusing very little on some aspects. For example, we've been only focusing on many detrimental effects due to heme toxicity that are associated with chronic hemolysis. However, as I mentioned in the beginning, a temporary hemolysis and the activation of this system in an acute phase might be also a positive factor that is really needed to reestablish homeostasis. And I recognize as a scientist, all these aspects are much less explored rather than toxic aspects. So you know better than me, Roberta, how much the products deriving from heme catabolism are important for protecting and for giving also beneficial effect. So we have learned a lot about chronic hemolysis, and I would like to see more studies exploring how acute condition require the activation of this heme-triggered mechanism, also for a positive and beneficial effect. Lately, it's been discovered that heme damage can also be associated to the transport of heme in vesicles also, that are released from red cell or from other cells. So this is an important new aspect, a new kind of variable we have not thought a lot about before. So I think new things may be coming on these aspects that change a bit the way we are used to look at heme, that we think as something going around in the circulation. So maybe these are novel aspects we should put more effort. But for me, finally, what is really coming down the long way of studying all about heme toxicity is now focusing on the therapies, meaning really we need now to answer the question after a lot of basic and translational research in mouse models that show that heme scavenging and preventing heme toxicity is important to for the pathophysiology of disease or marked by chronic hemolytic diseases, thalassemia and sickle cell disease. Now we have to be able to translate this in the clinic. And with really my great happiness, now studies and early clinical trials applying hemopexin in the clinic are ongoing. And I'm very much looking forward to see the outcome of these studies, because we have to be able down the line to translate what we have done into the clinic. And so I think this is really also the next step needed to understand, and also to go back to research to see what we still have to deepen, to understand, to explore in this specific topic and concept. Yeah.
Yes, I fully agree with you, Francesca. I think I've seen the studies you're referring to, the clinical studies. Perhaps I would add that because there are different mechanisms that are triggered by heme, perhaps there is the need of having multiple therapies applied together, hemopexin with maybe, I don't know, nitric oxide donors and
Exactly.
products of hemoxygenase activity that have antioxidant activity and so on.
I definitely agree. Combination therapy probably will be the winning one on the long run on patients that have such a complex pathophysiology, right?
Yes. So I had a wonderful time listening to your fantastic discoveries and your insights and you explained very well all the different aspects, especially related to macrophages and endothelial cells. I hope our listeners will also enjoy this dive into heme and its toxicity and the translational approaches that could be used in order to counteract this toxicity. And especially, I hope they understand the implication of this small molecule in diseases that are quite common in the population. So thank you very much for your time. I let you go back to your research. Good luck with all your work, and I look forward again to seeing your papers published and to learn more about this subject. Thank you, Francesca.
It was a pleasure. Thanks. Bye, Roberta.
Bye.